The malaria parasite is a single-celled microbe with a complex life cycle in two distinct organisms. Firstly, there's a vertebrate host like humans or mice, which is where malaria causes disease, and there's a mosquito vector which transmits the parasite between hosts. In the host, and indeed during most of its time in the vector, the parasite exists in a haploid state, meaning it has a single copy of its chromosomes and each of its genes, of which there's around 5,300. Here we only show two genes, A and B, for simplicity. In the first few hours after entering the vector, the parasite undergoes sexual reproduction when a male gamete and a female gamete merge into a single cell, which is then diploid and therefore has two copies of its chromosomes and each of its genes. This poses challenges for genetics and mutagenesis studies during the crucial early stages of parasite development in the vector. Any gene mutations that are introduced in haploid parasites within the host can then be rescued in these diploid stages in the vector by parasites that carry a non-mutated gene copy. But in this manuscript, we report a breakthrough that's helped us to overcome this obstacle. Based on our earlier finding that most male parasite genes are in fact inactive during the initial hours in the mosquito vector, we've devised a new approach. This involves conducting mutagenesis only in female parasites that would then be crossed with wild-type male parasites, resulting in unidirectional crosses and diploid parasites that carry mutated female genes and normal male genes. Since the male genes will be inactive, they won't then be able to rescue the function of these genes. And so, to achieve this, we had to use mutant parasites that produce only male or only female gametocytes. Figure 1b presents the results of these mutagenesis experiments using two different mutants that produce only female gametes. The first mutant showed poor survival in the mosquito, whilst the second mutant thrived. And so we selected that second mutant and developed the protocol outlined in Figure 1c. Here, a mouse is infected with the female-producing parasites, whilst another mouse is infected with male-producing parasites. We harvest the female-producing parasites and perform targeted mutagenesis on several genes of interest. Now, in this manuscript, we screened 31 genes, but this technique can be scaled up to include thousands of genes in the future. We then mix these two pools of parasites and infect another mouse. Once that mouse shows signs of infection, mosquitoes are allowed to bite the mouse and then ingest the parasites. Within the mosquito, the parasites undergo fertilization and then continue to develop as normal. At specific stages, we then harvest the parasites and use next-generation genome sequencing to identify which mutations persisted in the mosquito and which did not. Through this process, we've discovered 5 new essential genes for parasite development in the mosquito, which we characterize in this article. And so, to learn more about what these genes do, we invite you to read the rest of the article.